Today, it's a great pleasure to welcome Chie Aoki from New York University. And she's going to be talking to us a bit about the context of where she works and also about the effect of ketamine on animal models of anorexia. So without further ado, we'll just hand over to you, Chie.
Okay, thank you. Are you able to see the screen now?
Yes, we can. We can see
You
it
can,
nicely. Yeah,
you can see the screen and not my face?
that's
Or
right.
both?
Yeah.
Okay, that's fine. I would love for you to see the screen more than my face. But anyway, Sarah and Rupert, thank you so much for organizing this Ketamine International Journal Club. I've been a fan of the club for a few years now and learned so much from being exposed to the clinical side of research. It's especially important for me because my lab is located in New York University in Greenwich Village. It's a very cool place, but physically removed from the medical center. So I'm really looking forward to learning from this community as I share my newest findings, much of it unpublished. You say you can see the first screen. What I'm going to be talking to you about today is my usage of the animal model of anorexia nervosa called activity-based anorexia and some hopeful findings of using ketamine to ameliorate the maladaptive behaviors of ABA anorexia-like behavior. So anorexia nervosa, as almost all of you for sure know, is characterized by the symptoms of voluntary severe food restriction and low body weight. What some of you may not know is that the condition is also associated with excessive exercise. Some clinicians will say that almost all of their patients exhibit exercise that is excessive, and that even if there's a doctor's order to stop exercising, many of the patients cannot. It is an eating disorder that affects about 1 to 4% of females and about half as many of men. The onset is most commonly in adolescence, and it has an unacceptably high relapse rate, and this is true especially among adults. The mortality rate is very high, even surpassing that of depression, and there's at present no accepted pharmacological treatment. The outline of my talk today will be to mostly speak about the paper that was published this year in the International Journal of Eating Disorders, which reported on ketamine's ameliorative effect of reducing anorexia-like behavior in a model, which is a mouse, in adulthood. But I also will be talking to you today about the animal model of anorexia nervosa called activity-based anorexia, which began to enter the field in the 1950s using the rat model. I changed to make it a mouse model for my own purposes of wanting to introduce more genetically modified mice in the experiment. And I also would like to talk about the 2018 publication in the same journal, where we first reported on ketamine's ameliorative effect of reducing activity-based anorexia, but during adolescence. And a teeny bit about the mechanism of action of ketamine in reducing ABA vulnerability during adolescence. Some of the work just became accepted in publication last week. It's still in process of becoming the proof. So it's really, really very new. And if time allows, I also would like to talk about some limitations of the ketamine treatment when it's delayed into adulthood. And then how perhaps a combination of ketamine with ketogenic diet may influence a common pathway involving brain-derived neurotrophic factor and synaptic plasticity to ameliorate the maladaptive behaviors of anorexia nervosa, like behavior in mice. So what is activity-based anorexia? It does have the phenotype of very strong, excessively strong voluntary exercise, which comes about because the animal is starving. But the aspect of it that's interesting in its clinical, from the clinical point of view, is that these animals actually voluntarily choose to run rather than eat, thereby it being a voluntary food restriction that's manifested. And the combination of choosing to run rather than eat and exercising too much is what leads to the severe weight loss. How does this come about? We can make any wild type mouse or rat become anorexic by first simply acclimating the animal to a wheel. And for this, we just simply place a wheel in the cage and allow the animal to run as much as it wants, whenever it wants, inside its cage. We don't ever force them to go on the wheel. We allow the animal to acclimate for some 5 days, and then we introduce food restriction, which is to give them access to food only from 7 p.m. to 9 p.m. in the night hours, while keeping the wheel in there. And what we see is that within about a day, when the animals begin to lose body weight and become hungry, they run a lot more on that same wheel. And they're running, running, running. And if I were able to show you a video, you would be convinced that they appear abnormal in that they're compulsive in their running. And they choose to continue running on that wheel rather than eating the food that's right in front of them, even if their body is reaching 90%, 80% of their baseline, and they clearly, therefore, must be hungry. If I'm not careful, these animals will die. And so I have to make sure that I don't allow the animals to lose body weight more than to 80% of baseline, or else they will perish. So there are many symptoms of anorexia nervosa that can be captured in the mouse model. And we use this to quantify individual differences in vulnerability. So rather than looking at group differences, much of my data will show individual differences in vulnerability, either just naturally or in response to ketamine, and how that relates to anatomical changes. We see that the animals are clearly increased in their anxiety, and their increase in anxiety correlates very strongly with the extent of wheel running. So they're running because they are stressed. They're running because this is a foraging-like behavior, which is innate to the animal, which is hungry. And this is a behavior that's shared amongst all mammals that we have, or many people have studied. The severity of weight loss is more than seen by food restriction alone. And animals that are simply food restricted without the wheel will lose weight, but eventually learn to eat as much as they need during the 2 hours so they can regain and go back on their normal baseline and continue growing. But when they have the wheel, they cannot do that because they make the wrong choice of running instead of eating during those 2 hours. So again, the food restriction in the beginning is imposed, but they begin to show voluntary food restriction. We're able to use this model to also look at something like a relapse by presenting this ABA induction of wheel plus food restriction twice with recovery in between, and analyze propensity for relapse or the gain of resilience. We do see that some 80% of the wild type mice, if they are adolescent females, exhibit hyperactivity and severe body weight loss, but the vulnerability goes down to 50% when it's the second time around. It could be because it's the second time around and something of plasticity is evoked in the brain. We believe so based on our data that I won't have time to present today, but it indicates boosting of the GABAergic synaptic function in the hippocampus, prefrontal cortex, and amygdala. And those animals that continue to show this vulnerability show no plasticity of the GABA system. So the next question for us is, why are some animals exhibiting GABA plasticity while others are not? We don't have an answer to that. To be sure, there are symptoms that are not captured in the animal model of ABA, including dysmorphia. So in 2018, we were prompted by a study by Mills et al. In 1998, indicating that some of their his anorexia nervosa patients responded well to ketamine. But for some reason, since 1998, there were no other studies to follow up on this very intriguing finding of some 15 patients who were all long-term suffering from anorexia nervosa. What we did is to take animals that were in mid-adolescence and imposed ABA induction to them. We allowed the animals to then recover for about a week and then impose ABA again. To some of the animals, we gave ketamine during ABA while they were manifesting their maladaptive behavior of excessive wheel running and voluntary food restriction and beginning to show the first signs of weight loss. We gave them ketamine at 6 o'clock, 1 hour before their food availability. What we saw is that acutely within that day, food consumption went up, and that the food consumption improvement continued for the second and the third day of food restriction. Then in relapse, in ABA2, more than 12 days later, long after ketamine had cleared from their body, they showed a reduction of hunger-evoked wheel running, and that together with an improved food consumption led to better weight restoration as well as anxiety reduction. So since 2018, when we first observed this, we've been curious about how it is that ketamine might work, and more detailed analysis of their wheel running, which we can do throughout the 24-hour 24 hours of the day, indicated that what ketamine did with the administration just 1 hour before the 2 hours of food access, it was a reduction of food anticipatory activity in which was a sustained effect of ketamine. So this FAA, or food anticipatory activity, is a commonly used term amongst those of us studying activity-based anorexia nervosa. It reflects the finding that we've seen over and over across many laboratories, which is that animals can learn when they're going to be fed when they become restricted of food access. So for our animals, we feed them from 7 to 9 p.m. And then in the hours leading up to it, from 1 o'clock to 7 p.m., they begin to show more and more wheel running, from the first day to the second day of food restriction to the third day to the fourth day. Ordinarily, mice that are not starving are sleeping during the light phase, and they become active and roam around during the dark phase because they're nocturnal. But these ABA animals begin to show a huge amount of running during the day. And so that's the biggest change between food restricted versus ad libitum animals. And it is this time where we see the ameliorative effect of ketamine. So the ketamine dose we used were 0, 3, and 30 milligram per kilogram. For mice, the anesthetic dose is 100 milligram per kilogram. So this is clearly 30 milligram is sub-anesthetic. At that dose, when you do an IP injection, we see that the animals become a little bit wobbly for about 1 to 2 minutes. But long before 7 o'clock, they are doing just fine. You will not be able to tell the difference between animals injected with ketamine or with vehicle. They are able to run quite well. And so what we've begun to do in asking this question, how does ketamine work? 4 undergraduates in my laboratory helped me out with addressing this question, and we are having just a first inkling of how this could happen. We chose to look in the prefrontal cortex. Our favorite areas, as I told you earlier, were prefrontal cortex, hippocampus, and amygdala. And within prefrontal cortex, we chose layer 1. And we used the approach of electron microscopy so that we can look into the details of synapses in prefrontal cortex. We knew from the earliest time when ketamine was introduced into the field for veterinarians that ketamine is an open channel blocker of NMDA receptors. So we surmise that maybe NMDA receptors undergoing change in response to the blockade by ketamine. So we looked at the distribution pattern of ketamine at synapses, but what is good about electron microscopy is that we can readily identify synapses where there are presynaptic vesicles with a release of neurotransmitter, the postsynaptic spine where the receptors are expressed. Here the blue is indicating the NMD receptors and the red is indicating the AMPA receptor, all clustered facing the synaptic cleft where the neurotransmitters release. And these NMDA receptors are recognized to block these NMDA receptors. We also, by using EM, could differentiate NMDA receptors that are right at the synapse versus those that are tucked away in the cytoplasm. Why would they be tucked away in the cytoplasm? Well, the current literature indicates that with some synaptic input that would like to lead to long-term potentiation, there is an important reserve pool of neurotransmitter receptors right inside the cytoplasm near the synapse, but not on the plasma membrane, serving as a reserve pool for readily insertion of more receptors should they be needed. But kept inside so as to prevent excitotoxicity, excessive excitation. So we wanted to see whether NMDA receptors pool in the at the synapse or in the reserve pool could be changed by ketamine treatment in a sustained state of its looking at its effect. We also wanted to look at AMPA receptors because the literature derived from the depression literature using animal models had shown that ketamine has a sustained effect through desuppression of the mTOR protein synthesis, which leads to the increase in the expression of brain-derived neurotrophic factor. And with it, also increase in AMPA receptors, which are very helpful for allowing for new spines to form or the spines at the excitatory synapses of pyramidal neurons to become stronger, and is the basis for long-term potentiation. And so that's, going beyond the literature of depression or ketamine. We know the AMPA receptor reserve pool is important for enabling long-term potentiation or that form of synaptic plasticity. So we looked by electron microscopy and quantify the amount of these glutamate receptors at synapses and in the reserve pool, and then correlated them to behavior of animals that received either the efficacious dose of 30 milligram per kilogram versus the inefficacious dose of 3 milligram per kilogram. So we had 8 animals in these two groups and correlated to the behavior they had exhibited before we took the brains. And what I'm showing here is a correlation between the extent of NMDA receptor expression at dendritic spines of pyramidal neurons that are excitatory in the prefrontal cortex and of their behavior ante-mortem. So we had quantified how much they ran during FAA, the food anticipatory activity hours, during ABA1 and ABA2. And we looked at the difference to quantify the sustained ABA vulnerability in units of kilometer. So a negative value means that the animals gained resilience by running less during ABA2 compared to ABA1. Positive as shown in these two animal data points here, means that they actually ran more during ABA2 than ABA1. And 0 would mean that the animals stayed pretty similar during ABA1 and ABA2. So the blue dots are indicating the results of the correlation of the 30 milligram group. And we saw that there were 3 animals that gained resilience by running less. The Y value is negative. But the same animals were the ones that gained the most in the NR2B NMDA receptor expression. And there was a correlation for this group, very significant correlation. So even in the 30 milligram group, there were animals that did not improve in their food anticipatory activity, and then those that did, and there was a correlation between them. And there was no correlation for the 3 milligram group, which received the inefficacious dose of ketamine. Now, by EM, we were able to also look at the excitatory input to inhibitory interneurons. And for them, we found the same pattern. The 30 milligram group showed a strong negative correlation. So those that gained resilience by running less during ABA2 were the same ones that showed more NR2B immunolabeling at excitatory synapses formed on the inhibitory interneuron. So this is about the wheel running, and we found a correlation also with in terms of their feeding. So in this case, we want animals to be eating more, and those animals that ate more, as indicated by the y-axis, those animals that ate more were the same ones that gained more NMDA receptor at excitatory synapses. And there was this positive correlation this time. And that was true for the pyramidal neurons as well as for the inhibitory neurons. And so that difference in the correlation, negative for the feeding, for the running, and positive for the feeding, means necessarily that there are 2 pathways, parallel pathways, one for supporting feeding and another in the prefrontal cortex, another for supporting wheel running, and they're manipulated differently by ketamine. What we think is happening is that ketamine, by being the open channel blocker, will be blocking those channels that were belonging to pyramidal neurons that support the maladaptive behavior. And in that blockade of the synapses on neurons supporting the maladaptive behavior, then allows for the alternative adaptive circuit to become dominant in behavior. And that change happens over during the sustained, during the recovery period, and is manifested as sustained effect of ketamine. And we think that there is this kind of a pathway, parallel pathway in prefrontal cortex supporting change, whether it be of wheel running or in feeding behavior. And we have other results that we don't have time to talk about today, but indicating that indeed, in the prefrontal cortex, there are separate pathways supporting feeding, to feed more, or to suppress wheel running. So, about what I forgot to tell you is that the change we saw was mostly in the cytoplasm, that we saw the increase in the cytoplasm. So that means that with the gain of resilience, we were seeing increase in the cytoplasm more than in the synaptic junction. And that goes along with a previous finding we had indicating that animals with heightened level of NMDA receptor at the synapse were the ones that were vulnerable. So the gain of resilience was through the removal from the synaptic cleft just inside in the cytoplasm. Now, what could the cytoplasmic NMDA receptors? We also found that pattern is preliminary right now, but also for the AMPA receptor. What could this mean? I think it means that it keeps the pathways not too excitable. And that's good. Keeping it calm is good. It goes along with a increase in GABA tone that is helpful for a gain of resilience in terms of eating more and running less. But there's tucked away, just removed less than a micron away from the synaptic junction. So if need be, there's a way for NMDA receptors and AMPA receptors to be rapidly moved into the synaptic cleft in response to change in environment, such as stress. And we could get into the philosophy of whether this ABA vulnerability versus resilience, the need to run less and eat more, is really always adaptive. But I'm going to talk about that if we have time at the end. But in terms of sustaining its body weight for cure for anorexia nervosa, clearly the resilience means eating more and running less. So the challenge too, for me, was to ask this question about how ketamine might be helpful if given in late adolescence. And that's prompted by the understanding I had as somebody very close to patients with suffering from anorexia nervosa, that clinicians and families will probably hesitate using ketamine for treating the first onset of anorexia nervosa in mid-adolescence. But it is when we encounter the relapse of our loved ones that we really would like to know if there's a pharmacotherapy available. So I wanted to know whether during the relapse, if we were to treat with ketamine, whether that would be helpful. And in a publication in a chapter, I showed that actually it wasn't, that a single injection in late adolescence exacerbated the food restriction evoked FAA, the hyperactivity, and that exacerbated the weight loss. So what could we do? With the help of other undergraduates and a master's student, we were able to explore the possibility of helping in the relapse state with multiple doses of ketamine, as has been shown to be more effective in the human population as an antidepressant. So we tried 3 IP injections and during the relapse, and that turned out to be a good thing. We now were able to reverse what was exacerbated with a single injection. Now with triple injection, able to induce plasticity that brought in more adaptive behavior. So what we did in these experiments is to have the animals undergo ABA1 and then another relapse, and during that relapse, treat with ketamine, and then allow the animal to have a third ABA and see how whether there was a sustained effect. During the ketamine injection during ABA2, we saw an acute effect whereby animals ran less during the 2 hours of food availability. So this is the 30 milligram ketamine times 3 group compared to the vehicle group. There was a reduction in wheel running during the food hour, the 2 hours of food availability. And that's a good thing because that means animals are wasting less energy and using their time to eat more. And then during FAA of a third ABA is, so that's more than 12 days after the injection of the three injections with ketamine, we found that there was a reduction of food anticipatory activity amongst the ketamine group that was better than seen among the vehicle group. So each dot here represents data from one animal, and it's average across 3 days of food restriction during the third bout of ABA. Okay. And then now I want to talk to you a little bit in the language of correlation, see what we can learn from correlation. What we learned from correlation of which was really interesting to us. We saw that during the first ABA of this same population, there was a correlation between how much they ran from 1 to 7 p.m., and then how much they ate immediately following from 7 to 9 p.m. And this correlation was significant and negative. That is, the animals that ran the most were the same animals that ate the least in the subsequent hours of food availability. What we saw is that during ABA2, while the animals were getting the ketamine injection, the correlation changed from negative to positive only for the ketamine group. So now we were not able to reduce their wheel running, but those animals that ran a lot during the FAA from 1 to 7 were the same animals that ate more. Those animals that didn't run as much ate less. So they were able to now eat in a way that was more compensatory. They exercised, we couldn't help them from suppressing that, but those that ran more ate more under the ketamine influence. And very happily, this effect was sustained so that during ABA3, we still saw a positive correlation. Those animals that ran more ate more. In contrast, the animals that received vehicle, then during ABA3, they continue to show a negative correlation, which is paradoxical, but symptomatic as shared with the anorexia patients. That is, they exercise and continue to food restrict. Okay, so I'm going to skip this one and skip too much. Okay, and another correlation that we found interesting is that with all the animals, as I said, there was I didn't say, I'm going to say now, is that there's a trait that we can see for each animal in terms of how much they lost weight on the last day of food restriction at 7 o'clock just before feeding. So this is at the end of 3 days of food restriction and 22 hours since the last time they had seen a meal. Those animals in the black are those animals that receive vehicle. There's a strong correlation amongst them in that the animals that lost more weight during ABA1 were the same animals that lost a lot of weight during ABA3. Those animals that were relatively good about retaining their body weight during ABA1 were relatively good at retaining their body weight at ABA3. So there was something sustained in their natural behavior. But what ketamine did was to disrupt that prediction from ABA1 to ABA3. So, and they're in the prediction of gaining more during ABA3 than predicted based on how they were in ABA1. So this is my hero animal, heroine, because it's a female. It was one of the worst in terms of body weight loss during ABA1, but became one of the better in retaining body weight during ABA3. And I think this shift from the slope of correlation amongst the ketamine group indicates that they are able to depart from what was innate in them during ABA1. So then, so we are now into about 20 minutes into my discussion, and I now want to enter into unpublished work, which is to ask the question, can we treat ABA if ketamine treatment is delayed into adulthood? And the motivation for that is that relapse rate is the highest among adult females whose recovery from the first anorexia nervosa is incomplete. Will 3 times ketamine treatment help? So Sarah and Rupert, if this is a good time to answer questions about the published work, I can pause, or I can continue with presenting some data on this question of challenge 3.
Why don't you just carry on, Chiye,
Okay,
and finish? Thank you.
I'd be happy to do that.
Yeah.
Okay. So with that motivation, what we tried now is to see whether 3 times of ketamine could still be helpful if delayed into young adulthood. And to do that, we delayed the first onset of ABA in late adolescence, allowed them to recover, and looked at their ABA in the second into adulthood, and looked for how it relapsed again following ketamine treatment in adulthood. And we were first a little bit trepidatious about doing this experiment because the literature had said that these animals, the C57BL6 mice that are ordinary and not known to have anxiety traits, do not become anorexia-like. So what would happen if we did ABA in late adolescence? Do we even see anorexia-like behavior? In fact, we do. And in fact, they're actually showing more vulnerability when the first onset is in late adolescence. First, I want to say, let you know that it was disappointing in that weight restoration was not any better comparing ketamine 3 injection to saline. So looking at the weight gain during ABA3, there was a wide range of responses amongst the ketamine group indicated in red compared to the saline group. But if we look carefully, there was a less mortality. So if we use the same regimen of waiting till the animals are at 80% of body weight, of baseline body weight, to stop the ABA induction, and then that usually meant food restriction for 3 or 4 days, and you have to stop. And when the animals are older, this same regimen led to mortality of 2 out of the 11 saline group, which we did not expect, and but none of those who were treated with ketamine. And then here I want to show you a graph that's a little bit different from what I've shown before. We're looking detailed day by day of ABA1, ABA2, ABA3, and then within each graph, we're looking at the first day of food restriction, the second day, the third day, and the fourth day. What we saw that first with a vehicle group, 2 of the animals died during ABA1, and of those 9 that survived, none of them were able to retain their body weight to the cutoff of 80% or above. So here by ABA3, when they're really very mature adults, almost all of them had decline in their body weight due to food restriction without learning how to eat better or running less. While 6 out of the 13 ketamine group were able to start retaining their body weight. And here's a gray dot. Each color means a different animal. So 6 out of the 13 were able to gain resilience by losing less at 7 o'clock. Again, that's the last hour of food deprivation just before getting food. And then looking at their FAA, the wheel running leading up to it, we saw that the ketamine group showed some resilience, some 7 out of 13. We wish it was 13 out of 13, but it was 7 out of 13. But that's better than the vehicle group, where only one third of them showed resilience. And here again, we're looking day by day, FR1, 2, 3, 4 of ABA1, 2, and 3, and seeing that and using cutoff arbitrary of 1,000, 10,000 wheel counts per 6 hours, and 10,000 that's so in relation to kilometer, 32,000 is about 10 kilometers. So 10,000 is like 3 kilometers in 6 hours, which is a lot for a tiny mouse. And it's highly aberrant, and all animals show that. Almost all animals show that in the beginning, but some gain resilience during ketamine, and more so in a sustained way during ABA3. So there is some gain of resilience. And then if you look at ABA3 versus ABA1, which is before ketamine, more of the animals are able to show weight gain from 7 to 9 o'clock during ABA3 compared to the saline group. Unfortunately, when you compare the FAA correlating to how much they eat in the subsequent hours, as I showed you earlier, both the ketamine group indicated in red and the black indicating the vehicle group show there's still a negative slope. So we're still seeing that paradoxical combination of eating, not eating enough if you are of the type to run too much. And so this compelled us to think, what else can we do? And we were intrigued by the findings of a case study, Caroline study sort of described in the Frontiers in Psychiatry, indicating that when she combined ketamine treatment with ketogenic diet, she was able to feel the full recovery from anorexia nervosa that she had been suffering for since teenager years into adulthood. And that led to a pilot study by Calabrese et al. Of some 5 patients who were outpatients. So they were almost okay in terms of weight retention, but were still plagued by this haunting thought of needing to lose weight. And they, some 2 or 3 out of the 5 showed positive response to the combination of ketamine with ketogenic diet. And the literature had shown that ketogenic diet increases this brain-derived neurotrophic factor, and with that we could imagine a lot of synaptic plasticity that can be evoked by the ketogenic diet. It's the same diet that had been used for treating epilepsy. And so there is some history of its usage in the clinical setting, but I wanted to see whether my animals could respond to the ketogenic diet. And so there are 5 other undergrads at NYU who helped me with this study this summer. And the upshot is that it was amazing. Animals were much better in retaining their body weight. And because of that, their food restriction evoked hyperactivity, FAA was reduced. It's not that they were lazy, their total running was not that different, but the FAA was reduced remarkably. And with that, there was a reduction in mortality rate. So, and that can be shown here using the same kind of graph I just showed you, the body weight of animals day by day during ABA1, 2, 3 of those that received the standard diet versus ketogenic diet. Only 1 out of 9 of those animals that were receiving ketogenic diet had their body weight dip below the 80% cutoff during ABA3, while all of them who were on the standard diet were showing vulnerability by losing a very much body weight. And going along with that, their FAA, the wheel running among the standard diet animals, continued to show much vulnerability, 6 out of 9 showing vulnerability during ABA 1, 2, and 3, but those on the ketogenic diet, even though they showed some of them showed vulnerability during ABA 1, during ABA 2, only 1 show out of 9, and then 0 out of 4. We still have data coming in, which is the reason the denominator is less here, but are showing wheel activity. So you see like a flat line here. So we're really intrigued by this, and the future question is, how does ketogenic diet actually reduce ABA vulnerability? Is there some commonality with ketamine? And so far, the vulnerability is so scarce that we were ready to treat the vulnerable animals with ketamine, but we haven't found the need to do that. Okay, so that's everything that I was prepared to speak to you about today. And I would love to hear your questions and try my best to answer them. So thank you for your attention.
There's a lot of meat to chew on there. Thank you,
Uh-oh.
Jie. So please, members of the audience, please do put your questions in the chat, or put a hand up, or let us know that you want to ask a question. I'm struck by the by your methodology that seems to depend on the variability between the animals.
Yes.
When one's conventionally used to sort of thinking of these animals as being sort of rather homogeneous. Okay, is there something, can you comment on that?
I would love to. This heterogeneity was something I encountered from the very first day that I tried doing ABA, which is the reason that I'm thinking other people who were using the mouse model had said C57BL6 mice are not able to be used in an anorexia animal model. And so first I found the exception to that by using female adolescent mice only, while the field was mostly using male adults. And I thought, why we should be using female adolescents, because that's the vulnerable population. So let's look at that in the mice. And I was thinking that was the reason I could see it. But I think also I was always from the beginning interested in individuality, how it is that some animals and humans can gain resilience while others cannot, and trying to find a molecular explanation for that. So when I saw the individual differences, I welcomed it. But in the first litter that I tested, single litter, same mom and same dad, we found the heterogeneity in the single litter. We had 7 females in a litter. Some were very vulnerable and some were not. And so I started to think that maybe there's some differences in treatment, even before I start the ABA, some kind of experience, maternal pup interaction that brings about the individual differences, or maybe it's something in utero that I don't know about. But for sure, finding that heterogeneity in a single litter was really mind-boggling to me after many generations of inbreeding that these C57BL6 mice go through.
Can you say anything about how other people have fared with use? I mean, you've been at this for a little while now, so I imagine, have other people been interested and done it in rats or other models?
So most of the other studies that have moved from rats to mice have said that you have to use a particular strain that already shows anxiety trait to see a vulnerability to ABA. And so this is where I think they are not doing correlation analysis as I have, and that's where I think the departure is. If they were to relook at their own data, I think they will see what I'm seeing. And so by now I have had many students participating in these studies. And so different handlers, male and female. We have more females than male because I think my study attracts more females, but male, female, both, I see similar heterogeneity. I would say that most people have been, more and more of the other investigators are looking at female adolescents, as I have, and I think that's a good trend. But I must say that sadly the number of people studying the mouse model or the rat model is very small. I recently came back from the Society for Neuroscience meeting in Washington, D.C. It was attended by 20,000 neuroscientists from around the world, mostly Americans, but many of them, at least 30%, maybe 40% from outside of the US. And out of all of us, 20,000 presenters, there were 6 abstracts on this topic of anorexia nervosa, including human and animal models, and of them 3 were mine. So the field continues to be very, very small. And the reason for that is that the National Institutes of Health is not funding any research on this topic of anorexia nervosa. So recently my funding has come from private sources, and I was, advised not to acknowledge, but I really think it's important that these private foundations, Climate Foundation and the Vulnerable Brain Project Foundation, were very important in enabling me to do the research I've been doing in the past 10 years.
Can I ask you just a couple of questions about your data? It's like such a rich presentation, so it's wonderful to hear about your work. In terms of, how do you see this from a translational point of view, right? Because with the data you presented, it seems that, when ketamine is administered in a specific time in the life of the mouse, it can be beneficial, but, in adolescence can also be worsening the condition. So, and I think we can all agree that the lifespan of a mouse is not exactly the lifespan of a human. So how would you envision a translational approach on this topic?
Thank you for asking that, Sarah. I would say that my work has always been motivated by its value for translational research. So yes, that's why I showed the single injection working for mid-adolescence, but I found a way that it could work in late adolescence by boosting from a single dose to 3 doses. And but that's still in late adolescence. And even in adulthood, there is somewhat of an improvement. It's not 100%, but we're seeing more than half of the ABA animals responding positively to ketamine. So I think that what we're showing is that we have to be cautious that not overly optimistic that when ketamine is administered in adulthood, which is most of the case for as an antidepressant and probably will be for anorexia nervosa also, that it may not work for everyone. And what is the predictor for that? That's one of the important questions. For whom would ketamine be helpful? Can we know beforehand if it will be or not? But seeing that it can work for more than half of my mice, it gives me hope that if I were a mom of a daughter who was suffering from anorexia nervosa, I would seek ketamine therapy, I think. But I would also encourage a ketogenic diet. Now, one thing that was very interesting to me, well, initially my hesitance of trying ketogenic diet was because I felt that anorexic patients will have a hard time following the ketogenic diet because it, after all, is high fat, and anorexic patients will show fear to fried food or high fat. But it's also commonly known that ketogenic diet is a diet, that it's commonly thought that it is a way for reducing weight, which is the reasoning that these patients who were in the pilot study followed. They thought, oh, yes, it is high fat, but I only have to eat a little to reach the caloric need. And so eating small volume is, even if it's high fat, is easier than being forced to eat a big pizza, or in America, in the patient's ward, it's what they are told is to challenge by eating a huge bagel. That's like a big loaf of bread, or like three slices of bread, and not to be allowed to leave the table unless they ate a lot, but it's a lot of carbo. But instead of that, if they only need to take a few tablespoons of oil that they drink and regain their calorie that way and energy that way, to them, it seemed easier. So this ketogenic diet, I think, is not as difficult as I thought it would be for anorexic patients.
And on this point, and I think it was towards also the end of your presentation, right? Maybe still the slide we are seeing. When you tried the ketogenic diet on your animal model, that was not in combination with ketamine.
It was not.
It was
We
just
were,
the ketogenic
right.
diet.
We were prepared with a needle filled with ketamine, ready to go, and looking for those animals that continued to show vulnerability. So like this animal here is that, oh, here's 1 out of 9 that is continuing to show hyperactivity. But I can tell you that animal is now, I mean, I haven't included in the data point here, but that animal is not anorexic, not showing hyperactivity in ABA3, and it's not underweight either. So with the ketogenic diet alone, we've seen a lot of improvement. So I'm now busy comparing the BDNF level in the brain, in the prefrontal cortex, hippocampus, and striatum of the ketogenic animal versus the standard diet animals, and to seeing whether it's the sustained effect is brought up. Well, in this case, we can't say sustained because they're under ketogenic diet continuously, but whether there's a continuous elevation of BDNF in these animals, which is allowing for synaptic plasticity that reduces the maladaptive behavior. Oh, and the other thing, Sarah, that I didn't feel I had time to talk about was that we've seen using the animal model that there's an important sex difference. So I'm presenting to you today all the data from female mice, and the ketamine was not nearly as effective in male mice. And we're really perplexed by that. And I'm wondering whether such sex difference has been noted as an antidepressant. So I've begun to comb through the literature for that. But for my mice, it's we would need to maybe increase the sample size, but with a nearly equivalent sample size, we did not see as much of an improvement among the age matched, and they were actually from the same litter of male mice.
I'm not aware of any difference in terms of gender for the antidepressant response of ketamine, but you know, I leave this to the other experts that are here on this chat, at least among humans that I'm aware of.
Mm-hmm.
I can say that among humans, there is no difference in sex for antidepressant response. I have data from my clinic that's about to be published. So we've run through sex difference, age difference. It's the only one that we found out that older people it's harder to treat for depression, but only older people, not men or women, they are the same. And for anorexia nervosa, I think I had 3 patients, human patients. All of them were young girls with the same trouble eating, and they found out that maybe the ketogenic diet was a good choice because it's not that ugly food. That's how they interpret. They feel about the food, that thing in your mouth. It's not that, it's just some salad and some nuts and not that warm thing in your mouth. Maybe I can eat something like this, like avocado. It's not that bad for me. They think like that. That's the way they think. That I know it's more fat, but it's a good fat. It's not a bad fat. So they accept. Usually they accept. I talked to Lori Calabrese a few times about it. About the anorexia nervosa in humans and young girls. Most of them are young girls, and usually this feeling is shared that, you know, that hot food in my mouth, I cannot eat that. It's strange, but maybe some ketogenic diet with salmon or avocados or nuts or greens, they don't feel it's as heavy or as bad feeling in their mouth. And ketamine worked for them, not like a silver bullet. It's not one shot and they're eating hamburgers the next day.
Right.
It's not like this. It's a work of ketamine and psychotherapy and relationship with food, and it's a step, several steps, so they can get back to eat.
Yes. So I'm pretty close to Caroline of the who is the one person who is now saying she got cured by combining ketogenic diet with ketamine. She says that she will sometimes feel this bad thought about you must exercise, you must diet, returning, and when that happens, she goes on a ketogenic diet, and then that bad thinking recedes. So she hasn't had to return to ketamine treatment except once, which was about multiple doses, but one session. And that's really great to hear that they are listening to the voices and able to control it in some way. It's mysterious.
Lori Calabrese is an amazing professional, very enthusiastic with ketogenic and ketamine for anorexia nervosa. She's easily accessible and is an amazing person. And she has this program that you stay with the girls, with the women all the time, like they're in a house, like, and they all eat together and they all talk about it together, and they say that the headspace of dieting or exercising will be lower during the treatment. Not that you're not going to think about it, but your headspace. I will not only count calories or try to exercise or use my watch to see how many calories I've lost in the few hours, but the headspace will be lower. That's what usually happens. The girls I treated, they feel the same. My headspace is lower, so I can go to a party and I can have one thing or two to eat and not feel guilty on the other day or the same day, or not have to exercise to compensate that beer that I had last night. The headspace about it gets smaller. Very interesting.
That is very interesting. So you are already trying ketogenic diet and ketamine on your patients? I try
to push them to keto diet. They don't want to go. It's a very hard diet to follow for a long time, and they don't want to go. They just say, I'll try just ketamine. If it not works, if I'm losing it, I'll try ketogenic diet, because it's really hard to go to ketogenic diet and stay there for long. But they don't. They're not repulsed by the idea, like they are for regular food. I don't know if you can call it normal food and keto diet, but when say, okay, maybe I can try. That's what they usually say. Not like, no, absolutely not, I will not eat anything. No, they say, yeah, if I'm about to lose it, like I'm about to go on starvation, I may try it. I accept it, but I never reach that point. Usually they get better before it. It's a very hard diet. It's not easy to go.
Right. It's a relief to hear that one does not need to stay on the ketogenic diet, 24/7 across multiple years. You can go off and come back on it.
One thing that I teach them is that if you're not available to go to ketamine or any treatment, use keto diet as an emergency exit for the worst days. Like if you need it, you cannot go to ketamine. You just try keto diet, but don't stop eating. Don't stop the habit of having 2 meals a day. Switch it to keto diet, but don't stop the eating habit.
Yeah.
That's when they enter this stop eating and counting hours without eating, and they get competing with each other. I'm like 10 hours without eating. My top was 26. I would try 27, 30 hours without eating, and they starting competing with themselves how long I can be without eating or drinking. and how much exercise I can do it during this, and they start competing with themselves and with peers. It's not a healthy way to go, and sometimes they go to this, and I say, just do keto diet if you're in this situation.
Iago, thank you very much. And I'm afraid we've sort of run out of time, actually. And it has been an unusual journal club, because usually when we have sort of basics or scientists who are working at preclinical level, one doesn't get the sense of the sort of clinical almost case studies, and that's the sense that I've had with you, Chie. It's almost like we're talking to another clinician who's really interested in the variety and the heterogeneity that we face day in, day out. So you're among friends. You know, there's obviously a lot to disentangle here, but we're really grateful to you for coming and talking to us. I'd just like to finish up by saying something that I said right at the beginning for those who weren't here, which is that our website has changed, and so that you can now see all the recorded presentations, and also to encourage you to keep in touch and to think about coming to the conference that we're running on Oxford, 25th to 27th of March. So at that point, we're going to close the proceedings and just say thank you very much, Chie, for your really rich presentation and for the discussion afterwards. And we look forward to hearing more about this space. And it's fantastic that somebody's doing basic work in this area. So thank you for that.
Thank you so much again for this opportunity.